Hello friends and welcome. Today I am going to deliver a lecture on acute, short-term and chronic toxicities and its manifestations, mode of application, administration, exposure and in vitro tests. Let us first try to have a look at the main objectives of today's deliberation. To give a general and thorough idea about the acute toxicity, explaining in detail the short-term and chronic toxicities, to discuss in detail manifestations of drug toxicity, to give a general idea about the mode of application, administration and exposure. Finally, discussing in detail the in vitro tests and toxicity tests. Now we will start with the brief idea about the acute toxicity. Acute toxicity describes the adverse effects of a substance that result either from a single exposure or from multiple exposures in a short period of time, usually less than 24 hours. To be described as acute toxicity, the adverse effect should occur within 14 days of the administration of the substance. Acute toxicity is distinguished from chronic toxicity, which describes the adverse health effects from repeated exposures, often at lower levels, to a substance over a longer time period, months or years. It is widely considered unethical to use humans as test subjects for acute or chronic toxicity research. Otherwise, most acute toxicity data comes from animal testing or more recently in vitro testing methods and inference from data on similar substances. Limits for short-term exposure such as short-term exposure limits, STELS or ceiling value CVS are defined only if there is a particular acute toxicity associated with a substance. These limits are set by the American Conference of Governmental Industrial Hygienists and Occupational Safety and Health Administration based on experimental data. The values set by these organizations do not always coincide exactly, and in the chemical industry it is generally practiced to choose the most conservative value in order to ensure the safety of employees. There are also different values based on the method of entry of the compound, oral, dermal or inhalation. Threshold limit value, time weighted average, the maximum concentration to which a worker can be exposed every workday, 8 hours, and experience no adverse health effects. Short-term exposure limit, STEL or threshold limit value, short-term exposure limit, the concentration which no person should be exposed for more than 15 minutes during an 8-hour workday. Ceiling value, CV or threshold limit value, ceiling, the concentration which no person should be exposed to. When a person has been exposed to an acutely toxic dose of a substance, they can be treated in a number of ways in order to minimize their harmful effects. Obviously, the severity of the response is related to the severity of the toxic response exhibited. These treatment methods include but are not limited to emergency showers used for irritating or hazardous chemicals from the skin, emergency eye washes used for removing any irritating or hazardous chemicals from the eyes, activated charcoal used to bind and remove harmful substances consumed orally. This is used as an alternative to conventional stomach pumping. Now let us try to learn about combined drug intoxication. Combined drug intoxication, CDI, also known as multiple drug intake, is an unnatural cause of human death. CDI is often confused with drug overdose, but it is a completely different phenomenon. It is distinct in that it is due to the simultaneous use of multiple drugs, whether the drugs are prescription, over the counter, recreational or some other combination. The reasons for toxicity vary depending on the mixture of drugs. Usually, most victims die after using 2 or more drugs in combination that suppress breathing, and the low blood oxygen level causes brain death. The CDI-MDI phenomenon seems to be becoming more common in recent years. Elderly people are at the highest risk of CDI because of having many age-related health problems requiring many medications combined with age-impaired judgment, leading to confusion in taking medications. Now let us discuss chronic toxicity. Chronic toxicity, the development of adverse effects as a result of long-term exposure to a contaminant or other stressor, is an important aspect of aquatic toxicology. Adverse effects associated with chronic toxicity can be directly lethal but are more commonly sublethal, including changes in growth, reproduction, or behavior. Chronic toxicity is in contrast to acute toxicity, which occurs over a shorter period of time to higher concentrations. Various toxicity tests can be performed to assess the chronic toxicity of different contaminants and usually last at least 10% of an organism's lifespan. Chronic toxicity is the development of adverse effects as the result of long-term exposure to a toxicant or other stressor. It can manifest as direct lethality, but more commonly refers to sublethal endpoints such as decreased growth, reduced reproduction, or behavioral changes such as impacted swimming performance. Now let us discuss another part of our today's lecture, that is manifestations of drug toxicity. Many substances can cause harm when too much is taken, including alcohol, prescription and over-the-counter medications, illegal drugs, and some herbal remedies. The risk is increased when more than one drug is taken at the same time or the body is not used to taking a certain drug. An overdose occurs when a toxic, poisonous amount of a drug or medicine is taken. Substances that can cause harm when too much is taken, including alcohol, prescription and over-the-counter medications, illegal drugs, and some herbal remedies. An overdose is a medical emergency that requires immediate medical attention. A person's tolerance to overdose varies with age, state of health, how the substance was consumed, and other factors. The body often heals or without treatment. However, death is a risk in some cases. This may be instant or may follow more slowly if organs are permanently damaged. Treatment for overdose may be short-term or may involve ongoing treatment. For example, in case of self-harm or attempted suicide. General symptoms of a drug overdose may include nausea, vomiting, abdominal cramps, diarrhea, dizziness, loss of balance, seizures, drowsiness, confusion, breathing difficulties, internal bleeding, hallucination, visual disturbances, snoring deeply, turning blue, coma. Next is reasons for overdose. Taking an overdose may be accidental. A person takes the wrong drug or combination of drugs in the wrong amount or at the wrong time without knowing that it could cause them harm. Intentional misuse. A person takes an overdose to get high or inflict self-harm. Now coming to the risk factors of drug overdose. People of any age may take a drug overdose. The risk is increased when more than one drug is taken at the same time and the body is not used to taking a certain drug. First aid for drug overdose. If you think someone has taken an overdose, stay calm, call an ambulance on 000. If the person is unconscious but breathing, place them on their side in the recovery position. Make sure that the airway remains open by tilting the head back and lifting the chin. Check breathing and monitor their condition until help arrives. Do not try to make the person vomit. Do not give them anything to eat or drink. Bring the pill containers to hospital. Prevention of drug overdose. Some ways to avoid overdose include always read medication labels carefully and take prescription medications only as directed. Avoid drugs of any kind unless advised by a doctor. Always inform your doctor or other health professional of a previous overdose. Do not stockpile unnecessary drugs. Keep all the drugs and poisons at a safe place and out of reach of children. Be cautious when taking different drugs or substances. Now let us discuss the next part of our today's lecture, that is mode of application, administration, and exposure to drugs. A mode of action describes a functional or anatomical change at the cellular level resulting from the exposure of a living organism to a substance. In comparison, a mechanism of action describes such changes at the molecular level. A mode of action is important in classifying chemicals as it represents an intermediate level of complexity in between molecular mechanisms and physiological outcomes, especially when the exact molecular target has not been elucidated or is subject to debate. A mechanism of action of a chemical could be binding to DNA, while its broader mode of action would be transcriptional regulation. However, there is no clear consensus, and the term mode of action is also often used, especially in the study of pesticides, to describe molecular mechanisms such as action on specific nuclear receptors or enzymes. In pharmacology, the term mechanism of action refers to the specific biochemical interaction through which a drug substance produces its pharmacological effect. A mechanism of action usually includes mention of the specific molecular targets to which the drug binds, such as an enzyme or receptor. Receptor sites have specific affinities for drugs based on the chemical structure of the drug as well as the specific action that occurs there. Drugs that do not bind to the receptors produce their corresponding therapeutic effect by simply interacting with chemical or physical properties in the body. Common examples of drugs that work in this way are antacids and laxatives. In comparison, a mode of action describes functional or anatomical changes at the cellular level resulting from the exposure of a living organism to a substance. Route of administration. A route of administration in pharmacology and toxicology is the path by which a drug, fluid, poison, or other substance is taken into the body. Routes of administration are generally classified by the location at which the substance is applied. Common examples include oral and intravenous administration. Routes can also be classified based on where the target of action is. Action may be topical, enteral, but delivered through the gastrointestinal tract or parenteral. Routes of administration are usually classified by application location. The route or course the active substance takes from application location to the location where it has its target effect is usually rather a matter of pharmacokinetics. Nevertheless, some routes, especially the transdermal or transmucosal routes, are commonly referred to routes of administration. Pharmacokinetics, sometimes abbreviated as PK, is a branch of pharmacology dedicated to determining the fate of substances administered to a living organism. The substances of interest include any chemical xenobiotics such as pharmaceutical drugs, pesticides, food additives, cosmetic ingredients, etc. It attempts to analyze chemical metabolism and to discover the fate of a chemical from the moment that it is administered up to the point at which it is completely eliminated from the body. Pharmacokinetics is the study of how an organism affects a drug, whereas pharmacodynamics is the study of how the drug affects the organism. Both together influence dosing, benefit, and adverse effects as seen in PK/PD models. Pharmacokinetics describes how the body affects a specific xenobiotic chemical after administration through the mechanisms of absorption and distribution, as well as the metabolic changes of the substance in the body and the effects and routes of excretion of the metabolites of the drug. Next is the in vitro tests. In vitro tests and in vitro toxicity testing is the specific scientific analysis of the effects of toxic chemical substance on the cultured bacteria or mammalian cells. In vitro testing methods are employed primarily to identify potentially hazardous chemicals and to confirm the lack of certain toxic properties in the early stages of the development of potentially useful new substances, such as therapeutic drugs, agricultural chemicals, and food additives. In vitro assays for xenobiotic toxicity are recently carefully considered by key government agencies to better assess human risks. There are many substantial activities in using in vitro systems to advance mechanistic understanding of toxicant activities and to use of human cells and tissue to define human specific toxic effects. Most toxicologists believe that in vitro toxicity testing methods can be more useful, more time and cost effective than toxicology studies in living animals. However, the extrapolation from in vitro to in vivo requires some careful consideration and is an active research area. Due to regulatory constraints and ethical considerations, the quest for alternatives to animal testing has gained a new momentum. In many cases, the in vitro tests are better than animal tests because they can be used to develop safer products. Broadly speaking, there are two different types of in vitro studies depending on the type system developed to perform the experiment. The two types of systems generally used are static well palette system, the multi-compartmental perfused systems. Static well palette system. The static well palette or layer systems are the most traditional and simplest form of assays widely used for in vitro study. These assays are quite beneficial as they are quite simple and provide a very accessible testing environment for monitoring chemicals in the culture medium as well as in the cell. However, the disadvantage of using the simple static well palette assays is that they cannot represent the cellular interactions and physiologic fluid flow conditions taking place inside the body. Multi-compartmental perfused systems, new testing platforms, are now developed to solve problems related to cellular interactions. These new platforms are much more complex based on multi-compartmental perfused systems. The main objective of these systems is to reproduce in vivo mechanisms more reliably by providing cell culture environment close to the in vivo situation. Each compartment in this system represents a specific organ of the living organism, and thus each compartment has a specific characteristics and criteria. Each compartment in these systems are connected by tubes and pumps through which the fluid flows, thus mimicking the blood flow in the in vivo situation. The drawback behind the use of these perfused systems is that the adverse effects are more compared to the static systems. That's all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.